Hello, everyone, and welcome back to What's Next, the world of endotoxin. I am Oya.
And I'm Brendan. Today we have something very exciting. We'll be talking about one of my favorite topics, vaccines.
Yes, vaccines are very important and of great interest right now. But what do they have to do with endotoxins?
Well, Oya, the initial response to a vaccine can be similar to that of the primary response upon exposure to a pathogen, like endotoxin, as we discussed with Dr. Miller. But there is also a lot more.
Very interesting. So I would presume you have a great guest to talk about these topics today.
You know me too well. Today, from the Instituto Butantan, we have Patricia Canario.
Welcome, Patricia.
Thank you for having me.
Thank you for taking the time to be with us today. The Instituto Butantan is an amazing facility. Could you please give us some details of the institute and yourself, please?
Okay, of course. Butantan Institute was established in 1901, and it's one of the largest immunobiologicals and biopharmaceuticals producers in the world. We are famous for our collection of venoms, snakes, and spiders, and also for our antivenoms. But we produce 90% of vaccines and serums used in Brazil. And we have the highest production of influenza vaccine of the Southern Hemisphere. Nowadays, we also produce CoronaVac vaccine in partnership with Sinovac from China. And we are developing our own vaccine for COVID, besides one anti-COVID serum from horses. I am Patricia Carneiro. I am Quality Control Manager, and I've been working here in Butantan Institute since 2007, and always with production and quality control of serums and vaccines. I am veterinarian, actually, and I have a master's and doctorate in science with an emphasis of immunology.
Very impressive. So today, why are we talking about vaccines and endotoxins together?
So, like all injectable drugs, vaccine manufacturers must comply with strict quality controls for endotoxins. When vaccine antigen or protein carrier is derived from gram-negative bacteria, endotoxins are intrinsic in the product per se. In this case, they are part of the vaccine production and are considered as a process-derived substance. In that case, that can be used to monitor process consistency, characterize the endotoxin content in the product, and depending on the type of the product, can be associated with a pyrogen test to release the final product. When we have bacterial vaccines, we have this kind of situation. But we also have recombinant vaccines derived from yeast and viral vaccines made on viral cells or embryonated eggs. For this kind of vaccines, the endotoxin is considered a contaminant and should be monitored as a safety test for these products, complying with a specification from the compendium.
What are the characteristics of vaccines that can affect the detection of endotoxins?
In fact, the challenge that we face is not a sensitivity issue, but rather being able to have a good recovery of positive product control spike. The difficulty is intensified by gram-negative bacterial nature of some antigens or host cells, and sometimes the use of different treatments and purification steps during process development or optimization where formulation is not being set yet. Some constituents from raw materials of other components in the vaccine formulation may be present some characteristic of polysaccharides and can generate interfering beta-glucans with cause positive results.
So how do you guys use bacterial endotoxin testing?
Well, the vast majority of vaccines produced here at Butantan Institute are made from virus such as influenza, dengue, and vaccine against COVID-19 itself. Thus, they are products that not have the tendency for high endotoxin values. On the contrary, they are products where the presence of endotoxin is not common. For detection, we have in our quality control department equipment and material to perform 3 compendial tests: gel clot and the connective methods, turbidimetric and chromatogenic.
So does the Institute have plans to replace animal testing?
Yes, this is some excited topic here in Butantan. The Institute currently works in line with 3 Rs in order to minimize the use of animals, such as rabbits for RPT, use in low and intermediate states. The next step will be complete replacement using the monocyte activation test. But for all this change, of course, we have always regulatory challenges.
Regulatory challenges seem to be a lot. So thank you so much for your time today, Patricia. I, for one, will be the first in line as soon as my bracket is able to get the COVID vaccine here in Europe.
You had already COVID, Brandon. You have the antibody.
Maybe they're gone by now. But it's still important to make sure that everybody gets the vaccine, even if they've had COVID or not.
Yes. So remember, wear a mask, wash your hands, vaccinate, and stay safe.
Thank you, everybody.
Bye-bye.